Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 3 in our Openstax book. Chapter 3 covers these biological macromolecules and biomolecules. So all living things are made up of 4 classes of biological molecules called biomolecules. These include carbohydrates, we're going to talk about what those are, lipids, proteins, and nucleic acids. Now, these are the biomolecules, but only 3 of these 4 are what's known as macromolecules. To qualify as a macromolecule, you have to have large molecules that are composed of thousands of covalently connected atoms. So basically, this includes the carbohydrates, the proteins, and the nucleic acids. You know why? Because it's the carbohydrates, the proteins, and the nucleic acids that are made up of building blocks called monomers. And those building blocks called monomers, they link together covalently with covalent bonds to form polymers. Okay, the only one that doesn't do that from this list was lipids. So lipids, which include fats and oils and things, those molecules do not link together with covalent bonds to form macromolecules. So that's why they are not considered macromolecules. So again, the macromolecules are the carbohydrates, the lipids, the macromolecules are the carbohydrates, the proteins, and the nucleic acids. The lipids do not count. The lipids do not count as macromolecules, okay? The lipids do not count because they do not have monomers that link together covalently. Okay, so again, what is a polymer? It's when you take building blocks called monomers, you link them together covalently to form the polymer. Okay, let me break this down for you real quick. In carbohydrates, the monomer is called a monosaccharide. Okay, and the polymer is called a polysaccharide. Okay, I'm not going to spell the whole word, it's going to run out of space. In proteins, the monomer is called an amino acid. Okay, and in proteins, those amino acids link together covalently to form the protein. And another word for protein is peptide, right? Or polypeptide, actually. Polypeptide. Okay, and nucleic acids, that's your DNA, your RNA, right? The monomer is called a nucleotide. All right, a nucleotide. And the polymer, when the nucleotides get linked together, because the nucleotides are the letters basically of your DNA. You know, the A's, G's, C's, and T's of your DNA, and the A's, U's, C's, and G's of the RNA. They link together to form the DNA or the RNA. Okay, that's what you get when you link the nucleotides together. Okay, so notice that with fats or lipids, they do not have monomers that link together. They just have monomers. So if I have a fat molecule, it's just a fat molecule. I don't take one fat molecule and stick it onto another fat molecule and make some kind of polyfat molecule. It doesn't work that way. Okay, so hopefully that makes sense. Let's keep going. So how do you link these monomers together? You see, you link the monomers by this process on the left here called dehydration reaction, also known as dehydration synthesis. Look, you take the monomer. You see what I'm circling here? This is the monomer, and you link it to the polymer, the growing polymer, by how do you link them? There's actually a chemical reaction called dehydration reaction that happens, or dehydration synthesis. You see dehydration, an enzyme comes along and removes a hydroxyl group from either the monomer or the polymer, and a hydrogen from the other as water. You see an H, an O, and another H form H2O. So you're doing dehydration to make a covalent bond, and that's called dehydration reaction. And that's how you make the polymers. And it doesn't matter if we're talking about how you make proteins, you know, by linking amino acids together. It doesn't matter if we're talking about how you make sugars, you know, by linking monosaccharides together to make polysaccharides. It doesn't matter if we're talking about how we link nucleotides together to make RNA or DNA. It doesn't matter. In any case, in all of those cases, it's done by dehydration reaction or dehydration synthesis. You have to remove water to make the bond, and it's specialized enzymes that are doing that reaction. Okay, and remember, lipids do not have this reaction happen because you're not actually linking the lipids to one another. That's why they don't qualify as macromolecules. They qualify as biomolecules. Okay, and how do you break them back apart? So let's say on the right here, you see what I'm highlighting here. Let's say I have a polypeptide or a DNA molecule or something like that, or a polysaccharide. Let's say I want to break it back down into monosaccharides or amino acids. I want to break it down. How do I do that? It's the opposite. If I had to remove water to make a bond, I have to introduce water to break a bond. You see, it's just the converse reaction to the dehydration reaction. And this is called hydrolysis. Hydro means water, and lysis means break. You're using water to break this bond and release the monomer. So for example, if this was a protein, you'd do hydrolysis to release an amino acid from the protein strand. If this was polysaccharide like glycogen or something, you use water to break that bond and release glucose, the monosaccharide. For example, I'm just giving you examples. Okay, so again, sugars are your carbohydrates. These are basically the carbohydrates. Okay, sugars are carbohydrates because there's two real forms of carbohydrate, right? You've got the sugars and starches, but starch is just a type of sugar. So when I say carbohydrate, we're talking about sugars and starches. And remember, this is not only a biomolecule. Sugars or carbohydrates are not just a biomolecule. It's also a macromolecule because you have the monomers. The monomers are called monosaccharides. See, monosaccharides are monomers of sugar or carbohydrate, and they have the molecular formula. Okay, how do I know I'm working with a monosaccharide? It has a molecular formula of carbon. It has carbon, it has hydrogen, and it has oxygen. All right, sugars have carbon, hydrogen, and oxygen. And actually, they have, look, it says multiples. So sugars have one carbon for every two hydrogens for every one oxygen. So it's called a ratio. It's a 1 to 2 to 1 ratio of carbons, hydrogens, and oxygens. So for example, glucose is C6H12O6. All right, C6H12O6. Notice how there's 2 times the number of hydrogens as there is carbons or oxygens. And this is the most common monosaccharide, the most famous of the sugars, right? Glucose. Yeah, everyone's heard of glucose. It's a monosaccharide. It's a sugar, okay? But there's other sugars out there as well. For example, ribose is C5H10O5. Okay, C5H10O5. That's ribose, for example. You could even have 3 carbon sugars. It would be C3H6O3. Okay, so you can have sugars that are 3 carbon. You can have 5 carbon, 6 carbon. You could have 8 carbon sugars. But what do all the carbons? What do all the sugars have in common? They have a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. Okay, when you see that, you know you're dealing with a sugar. Okay, and by the way, if you want more explanation on this, I do have a link here that you can click and watch a video explaining this in a little bit more detail. All right, so actually, let me come back to these slides. Let's come back to these slides. Here's just an example of what I was talking about. Look, on the left, you have glyceraldehyde, which is a 3-carbon sugar. You see, you got 3 carbons, you got 1, 2, 3 oxygens, and you've got actually 6 hydrogens. There you go. That's glyceraldehyde. You also have ribose, which is a 5-carbon sugar. And you have here, remember we talked about glucose just now? Glucose is a 6-carbon sugar. And by the way, some of these sugars have, remember this group here? It's called a carbonyl group. Some of them have ketone carbonyl groups. These sugars are called ketoses. And some of them have aldehyde carbonyl groups, and these sugars are called aldoses. Although I'm not going to ask you about aldoses and ketoses per se, but it's good to understand that there's actually different structural isomers of the different sugars. In fact, let me show you some isomers here. You remember what isomers are? We explained that in the previous chapter, chapter 2. Remember, C6H12O6 doesn't always mean glucose. Why? Because isomers, right? Look at this. This first box here on the left, this represents glucose. Okay, this is a C6H12O6, right? That's what you think of when you think of glucose, right? But what if I told you galactose, which is this molecule in the middle? What if I told you galactose is also the formula is C6H12O6? And what if I told you fructose, which is the example on the right, what if I told you that's also C6H12O6? Well, you see, isomers are a real thing. Structural isomers are a real thing. You could have the aldose version of the sugar, that's glucose, see, with the aldehyde group, right? Or you could have the ketose version of the sugar, that's fructose, right? And then you have the cis or trans versions of, here you see, you've got the hydroxyl groups across from one another, so you would consider that trans hydroxyl groups because that's a geometric isomer. And then here you have trans here, but you got cis hydroxyl groups, but trans hydroxyl groups in glucose, cis hydroxyl groups in galactose. So all of these lessons we learned in chapter 2 about isomers is coming back to make a point here. So next time, if someone says, hey, do you know what molecule I have here? I have C6H12O6. Well, the smart thing wouldn't be to blurt out glucose. The smart thing would be to say, which isomer of C6H12O6 do you have? Do you happen to have glucose, galactose, or fructose? Okay, so just because you have a 6-carbon sugar doesn't mean you necessarily have one particular one. You have to figure out what the structure is. And by the way, you see how these sugars are drawn as a straight line? You see these sugars are drawn as a straight line here. By the way, when you add these sugars to water, they actually circularize. These sugars can become a ring. Okay, so this is what the sugar may look like outside of water, but you add it and look what the sugar does. Look what the glucose does. It actually kind of loops around and forms a ring. So you see this on the right here? This is the same thing. This is glucose, right? This is C6H12O6. This is glucose. Just like this is glucose. But this is the ring form of glucose, and this is the linear form of glucose. So don't get confused. Sometimes your textbook draws this and calls it glucose. Sometimes your textbook draws this other one and calls it glucose. So don't get confused. Usually glucose exists as a ring in solution. Okay, and look at this. This is really interesting as well. Look, you could even have 2 different versions of the ring form of glucose. You could have 2 isomers of glucose. Look at this. The one on top, the top right, this one's called alpha glucose. Why? Because look, you have the hydroxyl groups here, both pointing down. So that would be the cis isomer of alpha glucose, right? Now this glucose here, below it, notice how the hydroxyl groups are across from one another. This would be the trans isomer of glucose, beta glucose. You may actually want to learn the difference between alpha glucose and beta glucose, and I'll tell you why in a little bit. I'll actually explain why it's important to understand alpha glucose and beta glucose, these 2 different isomers of the ring form of glucose. By the way, here you have ribose. The ribose is good to understand too. Ribose is a 5-carbon sugar. Remember, it can also circularize. Remember this here, this 5-carbon sugar here called ribose. Well, this is the linear form of ribose, and then here you have it forms a ring. Here's the ring form of ribose, and fructose as well. Fructose can form a ring. So just because you see a ring doesn't mean it's not a sugar. There's definitely a sugar, and just because you see a straight line doesn't mean it can't be a sugar. As long as what? How do you know you're dealing with a sugar? You're usually dealing with a sugar when you have a 1 to 2 to 1 ratio of C's, H's, and O's. Okay, that's how sugars work. And then now you know there are rings, right? You form rings. That's called a monosaccharide, right? This is a monosaccharide of sugar. But how can I link those sugars together? Can I link them together? Look, here on the left, I have glucose, and on next to it, I have another glucose. And look, what do I want to do? I want to link this monosaccharide of glucose onto this monosaccharide of glucose. How do I link these two monosaccharides together to start forming a polysaccharide? You know, actually, first I make a disaccharide, which means two sugars stuck together, but then I can continue linking sugars on to make a polysaccharide. But how do I link them together? Do you guys remember how I link monosaccharides together, or how I link any monomer together for that matter? Well, remember this? Look, you have to utilize dehydration reaction or dehydration synthesis to remove water. That's how you make bonds. So here, look, you're removing a hydroxyl group and a hydrogen, and there you go. You've removed water. That was called dehydration reaction or dehydration synthesis, and you formed a covalent bond between the two monosaccharides. You formed what's called a disaccharide. That means two sugars stuck to one another, you see? And this bond here, you see how they're both coming down and sharing this oxygen here? Okay, this oxygen, actually this bond, I should say, this bond here, it has a name. It's called the glycosidic linkage or the glycosidic bond. So if I were to ask you what's the bond called between monosaccharides? It's called a glycosidic bond or a glycosidic linkage. And how did it form? Dehydration reaction or dehydration synthesis. Great. That's how it works. Okay, that's how it works. Now, how would I break this back apart? Before I say that, again, this is called a disaccharide because I have two sugars. If I were to add more sugars with dehydration reaction and make a really long chain, that'd be called a polysaccharide, right? Okay. So for example, if this was glucose and then this was glucose, I could link them together to form maltose, the disaccharide. But if I continued adding glucose on, I'd make glycogen or starch. Okay, so that's kind of how it works. Mainly glycogen. Now, how would I break this apart? How would I break this glycosidic bond apart? How would I break these two monosaccharides apart? Well, do you remember this back here? How do you break bonds apart? Oh, it's called hydrolysis. You're going to use water to break the bond. Let's see if we can do that. Look. Here's my disaccharide. Here's my glycosidic bond. And what do? Look, I take water, and enzymes do this, by the way. An enzyme would take water, attack that glycosidic bond, and look, the water actually becomes part of these two molecules. The hydroxyl group from the water is now here. The hydrogen is here. You've broken the two apart, so you've gone from maltose, a disaccharide, back to glucose, which are monosaccharides. Okay, so that's kind of how it works. That's how sugars are formed and broken apart in the body. Okay, and inside of your cells. Okay. This is showing you the same thing we just talked about. In this case, you're taking the monosaccharide glucose and you're linking it to the monosaccharide fructose, which remember both of these are C6H12O6, but this is an isomer. Glucose was an isomer. I believe it was the aldose isomer, and fructose was the ketose isomer. And you link them together with what? Dehydration reaction, right? And you form a glycosidic bond. So now you have glucose linked on to fructose, and that's actually called sucrose. That's table sugar. Okay, so take a look at this. So what's a polysaccharide then? Again, the polysaccharides are when you've linked several of these monosaccharides together. Okay, and what are two that you should know about? Two polysaccharides that you should know about? Starch and glycogen. These are two polymers or polysaccharides that you should know about. Starch is found in plants. It's how glucose is stored in plants. And glycogen. Glycogen is how glucose is stored in animals. And why would you want to store glucose? Well, because it's energy, right? You get energy from glucose. But let's say I have too much glucose to burn right now. Well, I could store it. And do you know where us humans store our glycogen, or a lot of animals store their glycogen? You know where we store that sugar? Mainly in our livers and in our skeletal muscle. That's where we store our glycogen. And in plants, we store the starch, or plants store the starch in little granules, in basically little vesicles. Okay. And by the way, both of these, of course, are storage of glucose. So it's a polysaccharide of glucose. Starch is a polysaccharide of glucose. Glycogen is a very similar polysaccharide of glucose. The only difference is really how they're linked together. You don't need to know the specifics about that. And then plants also have a different version of glucose linked together that forms what's called cellulose. And cellulose really doesn't have anything to do with making a polysaccharide for storage of energy, for storage of glucose. Instead, cellulose has a role in cell structure and stability. It's part of the plant cell wall, right? Have you ever bitten into a plant like a celery or broccoli or an apple? You know that crunch, that crunchiness? That's all cell wall material. That's cellulose. Cellulose, by the way, is also known as fiber, right? When you have a lot of fiber in your diet, they're talking about this cell wall material, cellulose. And again, plants link glucose together to form cellulose, but it's not to store energy, it's to give structural support to the cell. And of course, animals do not have cell walls, okay? Plants have a cell wall of cellulose. Plants have a cell wall of cellulose. Animals do not have cell walls, right? So you can see here those glucose linked together, and those glucose linked together form these really thick cell walls of plant cells. These are plant cells. So remember back here, real quick, let me flash back here. Do you remember back here I said you should know about alpha glucose and beta glucose? And I said I'll tell you why in a little bit. Why do you need to know that there are two different isomers of glucose? Alpha glucose, beta glucose? Well, here's why. Look. You remember I said there's starch or glycogen versus cellulose, and all of these are made of glucose? Well, here it is. Look, alpha glucose, the isomer is used to link together to make starch and glycogen. Okay, for energy storage. Beta glucose, remember the trans version of glucose, is linked together to form cellulose. So even though starch, glycogen, and cellulose is basically a polysaccharide of glucose, there's a fundamental difference, and that fundamental difference has to do with alpha glucose being used in starch and glycogen versus beta glucose being used in cellulose, okay? So if all of these are made out of glucose, right? If starch, glycogen, and cellulose are made out of glucose, then how come when I eat starchy things like potatoes, or if I eat a lot of meat which has a lot of glycogen, if I eat a lot of glycogen or a lot of starch, I gain a lot of calories and I can gain weight easily, right? But how come if I eat cellulose, a bunch of what's called fiber, how come I don't gain any weight? How come I can't even digest it? I can't even digest the cellulose. What's going on with that? Well, here's what you need to understand: the power of isomers. Look, you see alpha glucose? When alpha glucose is linked together, the strands look like this on the left, and all the hydroxyl groups are cis. They're pointing down. Okay. Your body has enzymes that can break this down, like amylase, I should say, for example. You've got enzymes in your digestive system and in your mouth that actually can break down sugars. Especially the alpha polysaccharide. Okay, the alpha glucose polysaccharide. However, your body does not have the appropriate enzymes that could recognize this, that could recognize cellulose. So you don't have an enzyme when the hydroxyl groups are trans like this or staggered like this. You don't have an enzyme that recognizes this and can break down the sugar. And if you can't hydrolyze, remember hydrolysis, like breaking these polysaccharides back down into monosaccharides. If you can't break these polysaccharides back down into monosaccharides, you can't digest it, right? So think about that. Think about that. You can eat celery all day long, and really you're not gaining a lot of calories. Why? Because even though you're eating a ton of cellulose, which is basically pure sugar, it's pure glucose, right, in the cell walls of celery, you're actually not gaining a lot of calories because, again, why? You can't take that celery and break it back down into glucose. You don't have the appropriate enzymes. Whereas if you ate a bunch of potato chips, which have starch, well then, yes, you could break the starch down back into glucose, burn the glucose, and every time you burn glucose, you're getting energy, and you can, and if you get too much glucose, then you start storing it as fat, right? So you gain weight. Okay, so that's all explained here. What I was just talking about is all explained on this slide. Okay. So before we move on past carbohydrates, move on to lipids, which would be the second part of this lecture. Let me take a quick break. Maybe go get a glass of water, and I'll be right back. So it's break time. Hey everyone, welcome back from break time. We're just about to jump into this concept of lipids. Lipids is another type of biomolecule, but remember what I was saying earlier, lipids are not macromolecules. Why? Because lipids do not link together to form polymers, okay? They stay monomers. They do not link together. And there's many different kinds of lipids. It's quite a diverse group. They include the fats, okay, phospholipids and steroids, which we're going to talk about, and also oils, waxes, and some of the vitamins as well. Okay, all of these fall under the umbrella of lipids. So because it's such a diverse group, one thing I will tell you that actually unifies, it's a unifying characteristic of all of these is that they're hydrophobic. Okay, remember, do you know what hydrophobic is? I don't think I've used that word yet, but hydrophobic means water-fearing, okay? That's just another fancy way of saying non-polar, right? You remember non-polar molecules? They don't have any charge, right? So do non-polar molecules mix with water? No, non-polar molecules don't mix with water, right? And that's because they don't have any kind of charge. They need at least some kind of partial charge or a charge or something to mix with water, remember? So things that are non-polar don't mix with water. And another way of saying that is hydrophobic. Hydrophobic molecules are non-polar. They don't mix with water, okay? So what kind of molecules did we learn are hydrophobic? What kind of molecules did we learn don't mix with water? Well, what about this right here? It tells you hydrocarbons, right? Big old molecules that are made of hydrogen and carbon. Those molecules, they're going to be non-polar. They're not going to, because why are they non-polar? They're non-polar because of non-polar covalent bonds. They're not going to want to mix. They're not going to want to mix with water at all. Okay. So again, one thing that fats, phospholipids, steroids, oils, waxes, vitamins, not all vitamins, by the way. There's like the ones that are actually fats. What do they have in common? they are mainly hydrocarbons, right? They're mainly long chains of hydrogen and carbon and very non-polar, right? So for example, look at this molecule at the bottom right. Look at the bottom right. That is a fat molecule, also known as a triglyceride. Okay, fat molecule. And how do you form this fat molecule? You take this part here on the left, the bottom left. This is called the headpiece, or glycerol. See, glycerol is the headpiece. And onto the headpiece, you link 3 of these tails. These are called fatty acid tails. See, right here. This is called a fatty acid tail. 3 of these fatty acid tails link onto one glycerol headpiece to form a 3-tailed molecule called triglyceride. Okay, this is a fat. This is a fat. Okay, and is it any wonder that this molecule does not mix with water? Look, it's essentially a giant long creature with 3 legs, and all of its tentacles here are long chains of carbon with hydrogen attached, right? So that's not going to mix with water, that's for sure. The bunch of hydrocarbons, okay? Even though there's a few oxygens here and there, that's not sufficient to make this a molecule that can mix with water. This molecule is by no means going to mix with water, okay? Fat molecule. This is why oil and water don't mix, or fat and water, butter and water, for example, don't mix, okay? They're just not going to mix. And by the way, let me show you something really interesting. This molecule here, it's called fatty because it's got a bunch of carbons and hydrogens, and that's essentially what fats are. Why is it called an acid? Just quizzing you real quick. Why is it called an acid? We know why it's called fatty. It's called fatty because it's got long chain of carbon and hydrogen, all right? Why is it called an acid, right? Think about that. Maybe pause this video and think about it and try to answer it on your own for a minute before I give you the answer, okay? You ready for the answer? Well, look at this. What is this? What is this right here? What am I circling? Isn't that a carboxyl group? A carboxyl functional group? Do you remember in chapter 2 I said when a molecule has a carboxyl functional group, it's technically a acid, right? So anyway, now you know why this is called a fatty acid, because it's basically an acid because it has a carboxyl group attached. However, when you link the fatty acid onto glycerol, you kind of destroy that carboxyl group. And now, with the help of little ester bonds, which you don't need to know, you've got a fat molecule. Okay, so a fat molecule has a glycerol head and 3 fatty acid tails. Okay. Here's another thing you should understand. You remember the fatty acid tail we were just talking about? You could have just single bonds between the carbons, or you could even have sometimes double bonds in the carbon tail. Look, this is C double bond to C, carbon double bond to carbon. You see that? So do you see any of those in this example here? No, in this example, it looks like just single bonds, right? But you could have a tail that has a double bond in it, okay? And here's why that's important. If you only have single bonds in the tails, that's called a saturated fatty acid. And if you have a double bond, even one double bond anywhere, it's called an unsaturated fatty acid. Does that make sense? So what's this fat right here at the bottom right? What's that? Is that a saturated fat or an unsaturated fat? That's a saturated fat because I don't see any double bonds between the carbons and the fatty acid tails. You see that? So why is that important? Well, but before I tell you why that's important, which one has more hydrogens? If you have a saturated fatty acid tail and an unsaturated fatty acid tail of the same length, which one would have more hydrogens? Think about it. Which one would have more hydrogens? Well, obviously, yes, the saturated fatty acid has more hydrogens because look, by making that double bond in the carbon, you're using up 2 bonds. So you can only have 1 carbon attached to each of these carbon here. You can only have 1 hydrogen. So you're actually going to end up with fewer hydrogen on unsaturated fats than you do on saturated fats. So that's good to understand. So why is that important here? here's a saturated fat example on the left, and here's an unsaturated fat example on the right. So, by the way, do you see this right here? Do you see this molecule? Can you tell that this molecule is actually this? Okay, because think about it. How would I draw this? How would I draw this fat, right? If I was an organic chemist, do you think I would want to draw this whole fat molecule out, right? Would I want to draw all these carbons and hydrogens? Oh, no way. Let me show you something. Okay, do you guys remember I taught you how organic chemists do shorthand for things? Okay, so instead of drawing this giant molecule, you know what an organic chemist would do? They would draw glycerol like that, and then they would say, well, glycerol branches into, I don't know, an oxygen, right? And then that goes to a carbon and to another oxygen, right? Is this correct so far? I think so. And then guess what? Zigzag tails, right? So check this out. I could just draw a zigzag tail because do you remember each one of these vertices, each one of these little points is a carbon, right? So for example, that point is a carbon. And how many hydrogens would it have? 2. So I could sit there and fill in. Every carbon and every hydrogen and every. I've already written in the oxygen. So just by drawing this little zigzag, what looks like a zigzag diagram, like almost like what football players would use in a play. By drawing this, I've actually saved a bunch of time and not had to draw this here. You see this. And put in all those carbons. So that's what they did here. See, remember, this is where that shorthand that I taught you earlier in chapter 2 pays off, right? Because look here, this molecule is this, okay? But do you see any double bonds in the tails? No, it looks like only single bonds, so that's a saturated fat. Do you see a double bond in these tails? Yeah, look, there's a double bond right there, And because it has that one double bond, it's called an unsaturated fat. So if you have even one double bond, it's an unsaturated fat. And again, unsaturated fats would have fewer hydrogen than saturated fat. We already talked about that. And look what it causes, the double bond. Look what the double bond causes. It causes a kink. It causes a kink in the tail, look. And do you remember when carbon, we talked about this earlier too. Do you remember when carbon makes a double bond, it becomes flat, like planar? That's kind of what's causing the kink, by the way. You're going from tetrahedral shape to planar shape. Okay, so what's all this important? Again, saturated fats only have single bonds in their fatty acid tails. Unsaturated fats have at least one double bond. Saturated fats have more hydrogen than equal sized unsaturated fats. Here's another thing. Saturated fats tend to be found in animals. Unsaturated fats tend to be found in plants and some fish. Okay. Here's another thing. You might ask, well, how come animal fats tend to be solid, like butter or lard, right? And how come plant fats tend to be liquid, like olive oil or vegetable oil? How come saturated fats are solid at room temperature usually, and unsaturated fats are liquid at room temperature usually? It's the same temperature. How come one's solid and one's liquid? Well, the best example I have for that is here. Take a look at this. You see how you have 3 tails? Well, if they don't have any kinks, if it's just 3 straight tails, when the temperature cools down, these 3 tails have an easy time kind of resting on top of one another and stop moving, right? That's what freezing means. That's what turning solid means. Like, so when butter turns solid, basically these tails stop moving. And it's easy to stop moving if you're straight, right? Okay, but if you have a little kink in your tail, if you have a little kink in there, that could cause slippage, right? That can cause the tails to keep moving, right? And what do unsaturated fats have? They got a kink in the tail somewhere. So when the temperature cools down and these fatty acid tails try to stop moving, and so the thing freezes or becomes solid, this kink can cause this tail to keep moving, and that's going to keep everything liquid. Okay, so that's my best explanation to why unsaturated fat is liquid and a saturated fat is solid. Okay, so that's good to understand. We already talked about this. Okay, great. Another type of lipid to talk about that we're going to discuss is called a phospholipid. Okay, this is not a fat. It's a different kind of lipid. It's called a phospholipid, and it looks kind of like this. Okay, and the reason phospholipids are important, by the way, is that they make up the membranes of your cell. You remember when I said your cells have a membrane around them, and the nucleus of eukaryotic cells has a double membrane, and there's a membrane. There's membrane-bound organelles like the mitochondria and chloroplasts have membranes around them. Every time I say membrane of a cell, this is what it's made out of. Phospholipids is what membranes are made out of, right? And phospholipid, one unit of a phospholipid looks like this, okay? Phospholipids have, basically, look at this real quick. Phospholipids kind of look like a fat, don't they? You see this structure right here? You know what this is right here? This structure right here. That should look familiar. It's glycerol. Remember this guy? Here, glycerol. Do you remember what a fat is? A fat is glycerol with 3 of these guys, right? 3 fatty acid tails, right? That's a fat. Glycerol, 3 fatty acid tails. What's a phospholipid? It's almost the same thing. Look, it's glycerol with 2 fatty acid tails instead of 3, okay? Instead of 3 tails, the only difference is you got 2 tails with the glycerol, okay? But in addition, it's called a phospholipid because you have a phosphate group, and the phosphate group's charged, and then usually you have another charged group as well. You see this little R up here? That's another little extension. Usually it's something like choline, which is also charged. By the way, it's charged, okay? So basically, what is a phospholipid? A phospholipid is the main component of the membrane of the cell, any kind of membrane around the cell or in the cell. And it almost looks like a fat molecule, except instead of glycerol and 3 fatty acid tails, you got glycerol and 2 fatty acid tails. This appears to be an unsaturated fatty acid, by the way. And you have a little phosphate group that's charged. We talked about phosphate groups in chapter 2. Remember, it's one of the functional groups we learned about. And then another little group here. So why is that important? Because look, isn't phosphate group charged? It's a negative charge phosphate group, see? Doesn't that mean water can mix with the phosphate group? Remember, water can mix with anything that's charged, and isn't phosphate group charged? So water will actually mix with this part of the molecule. This is why it's called the hydrophilic head. You see this round circle here? This, you see this up here? This here is a hydrophilic head. Okay, the hydrophilic head. Hey, Tig. Okay, and the hydrophilic head, this is my cat Tig, everyone. The hydrophilic head is actually, hey, how are you? Say hi. Sorry. The hydrophilic head is actually, it mixes with water because it's got the charged phosphate group on it. Okay, it's got a charged phosphate group, and phosphate groups can mix with water. That's why this part of the molecule, this circle here, is called the hydrophilic head. But what about the tails? Remember what the tails are made out of? The tails are actually made out of long chains of hydrogen and carbon. Here, check them out. Hi. Hi, Tig. His name's Tigger, by the way, like of Winnie the Pooh fame, in case you're wondering. He's come to say hi. He gets lonely sometimes. Yeah. Okay, I'm sorry. I'm getting sidetracked here. I'm getting distracted with little Tig. Hold on. All right, I'll get right back into it. Hi. He's the best cat ever. Anyway, notice the tails here, down here, right? The tails would not want to mix with water, would they? Okay, because they're just long chains of carbon and hydrogen, aren't they? So these tails are not going to want to mix with water. That's why the tails are called hydrophobic tails, okay? And how does this make a membrane? You're like, this doesn't look like a membrane at all, right? Well, it looks like, so you've got the head with two tails. That's just the basic unit of the membrane. You see, so the membrane actually looks like this. You've got phospholipids, which is each one of these heads with the two tails. That's a phospholipid. Here's another phospholipid. Here's another phospholipid. So there's a row of phospholipids with the heads pointing outside of the cell, and then you would have a row of phospholipids with the heads pointing inside of the cell. Does it make sense why the heads? First of all, you have what's called the phospholipid bilayer, right? Bilayer means two layers of phospholipids. See, here's the first layer of phospholipids. Here is the second layer of phospholipids, okay? The first layer has the heads pointing outside the cell. The next layer has the heads pointing inside the cell. You know why? There's a lot of water outside of the cell, and there's a lot of water inside of the cell. And the water can mix with the heads, right? No problem. The water can mix with the heads. So what are the tails doing? The tails are kind of sequestered in the middle of this zone. And this zone here where the tails are is called the hydrophobic zone. That's where water does not exist. There's no water in this where the tails are. That's a completely water-free zone. It's called the hydrophobic zone. Okay, so phospholipids form a bilayer, what's called the phospholipid bilayer, with the heads, the hydrophilic heads pointing outside the cell and inside the cell where the water is, and the tails are all devoid of any kind of water, and they're in this hydrophobic nonpolar area of their own, okay? And things can't just cross the membrane. It's actually like a little force field. Things can't just randomly cross the membrane unless they have special properties or they have a transporter to transport them across the membrane, right? So it keeps the stuff inside the cell inside, and it keeps the stuff outside the cell outside. Even water can't just cross the membrane. Let's say you're a water molecule. You see my red dot here? Imagine if my red dot is a water molecule. Even if a water molecule wanted to go and cross the membrane, it would get bounced off, right? See the water molecule trying to go in? Boom, it would get bounced out. Because all of these tails are hydrophobic. It's almost like a hydrophobic force field, right? Last thing to talk about in Chapter 3, Part 1, this is the last concept of today anyway, of this video, is steroids. What are steroids? Steroids are another kind of lipid. You know, lipids so far, we've talked about fats, we've talked about phospholipids. Steroids is another kind of lipid. Notice how it's a bunch of carbons and hydrogens, but look, it's 4 fused rings of carbon and hydrogen, right? 4 fused rings. 4 fused rings. Where have we seen 4 fused rings before? Where have we seen 4 fused rings? Maybe in Chapter 2. Remember estrogen or estradiol and testosterone? Those are steroid hormones. Those are also steroids. Those are steroids as well. But in this example, you have cholesterol and cortisol, right? 4 fused rings. So in your body, your natural hormones in your body, a lot of them are steroid hormones, okay? And they have 4 fused rings. But what makes one steroid different than another steroid? Do you remember what made estrogen different than testosterone? What's making cholesterol different than cortisol? It's the functional groups, right? It's what is attached to your carbon backbone, right? What is attached to your carbon backbone? Now, you don't have to memorize this one's cholesterol and that one's cortisol and this one is estrogen and that one is testosterone, but I do want you to know that steroids are lipids and they consist of 4 fused rings. Okay, and what we're going to do next time, in Chapter 3, Part 2 is go over proteins, okay? So we'll start on this slide for the next video. Okay, thank you again for watching. I hope you learned something. Let me know, ask a question, and as always, take care till next time.